Hi everyone. I'm sorry. Hi everyone. I'm gonna present one of my ongoing projects at Holden Arboretum, and this is called From Petals to Pests: The Multifaceted Role of VOCs in Rhododendron Species. So to start with, I'm gonna explain a little bit about these VOCs that stand for the volatile organic compounds, and they are basically these smells that the plants can emit. And then we thought that they were just from flowers, but we already know that they are also from leaves, and they have different functions that are important, especially for pollinators. So one of the functions of those VOCs are the attracting pollinators. That's one of the functions they have, but they are also good for kind of like microbial attraction, and also like they can repel them, as well as they can attract and repel different pests. And I have an specifically example here. So on the right side, I'm showing one example with one VOCs, and they are called herbivore-induced plant volatiles. Those are VOCs as well, and they have found that, for example, we have at the bottom we have this rhododendron lace bug, which is one of the pests from rhododendron species, and they have found that with the plant know that they are like kind of like threatened by these bug or like these pests, they can emit different like VOCs to kind of like let other plants know what is happening to the actual plant. But also these VOCs or these HIPV volatiles can help the plant to attract also like different predators for the pests. So this is really interesting, thinking about the vulnerability and also the conservation of these species, and how these VOCs or these specifically the HIPV can attract those predators that can reduce the pest populations in different rhododendron species. So we already know that there is like around like 1,200 species worldwide, but most of the work on rhododendrons, specifically on VOCs, have been focused in the Asian area. And I'm showing here this map, and this is just to show kind of like this is the diversification rate, but I'm trying to kind of like picture the distribution of rhododendron species in the worldwide, and focusing on how like the North American species, the native North American species that we have, are understudied, specifically in the VOCs perspective, and how important it is for us to understand, like first which VOCs those plants has, but also what are the implications and the importance for the species that we have right here. So based on that, I have this aim, and this is to characterize the VOCs from leaves and flowers of different North American Azalea species. And I think I mentioned, but I just want to mention that again. And it's this is an ongoing project. I have just like a preliminary data to show today. So we are focused in different North American species, and that we have at Holden Arboretum. And also I'm going to be focused in the three last ones that I highlight here are the species that present like a higher or like strong smells. So those are kind of like we expect that they will like show us like differences between those species and the other species that we have. For the VOCs analysis, we have a collaboration with Gardner Lab at Case Western Reserve University. So what we're gonna do is collect those samples from the flowers and leaves. So we are gonna like randomly select three inflorescence and 3 leaves aggregations. We are gonna extract those VOCs using a vacuum pump, and kind of like take them and put them in like seals tubes that we can analyze using a GCMSS that Dr. Gardner is gonna help us with. And after that, we're gonna analyze those results or those data that we will have to establish which kind of like VOCs we will have from the samples. We already did like a preliminary analysis from Rhododendron oldhami, and we found, I'm just showing here two of the VOCs that we found, and they are like correlated with those, one, the odors, like the grays, like grass, green odors, but also with this kind of like function when they can attract those predatory insects that I mentioned at the beginning. But also we found another volatile that is like highly related with the VOCs from rhododendrons. This is what we have so far. We're hoping to have more samples next year, the like peak flowering from the rhododendrons that we have in the garden. But we are really excited to keep working on this project that is ongoing. On the other side, I want to show about a little bit about another project I have going on, and it's kind of related with the VOCs as well, but it has kind of an independent perspective as well. And one of them is like evaluating phenology of different plants. And this phenology is kind of the cycle that we can evaluate from the whole, like for the plant. And I'm showing here like a picture of different like phenophases. Those phenophases are like the phenology phases. But when we're thinking about rhododendrons and other plants specifically, it gets a little bit more complicated because we have like so many, or like more phenophases that we can evaluate, right? So we are really interested in this because this can be related also with environmental conditions. And for example, if the spring gets warmer, we're expecting that the flowering season is going to be earlier. And this is really good for the research perspective in order to get our data, but also in terms of horticulture at Holden, to know like different processes that they do have depending on this phenology. Also, we're interested in the pollinator perspective and how those pollinators are being attracted. And this is where the VOCs are kind of like convert, like they like join with the phenology part and this association with the rhododendrons. And we are like wondering what is like attracting those pollinators, if that's more the VOCs or like the different traits that the flowers might have, like color or like the pigments that that other team like lab mates are working on, or it might be the size, it might be the shape, it might be like different other associations that might be affecting that, or might be driving that pollinator attraction to the plants. So based on that, we are aiming to describe the pollinator visitors and the phenology of the plants involved in the study based on flower morphology and field observations. And to do this, we did have two options, and one of them were like go in the field and take the data, which is good for us. But we wanted to start like a long-term project when we have really like, you know, like more robust data and a like more like closer and more detailed kind of monitoring in the gardens. And at Holden Arboretum, we have like a really strong volunteer program. And that's why we decided to start a long-term volunteer program, which is called like Rhododendron Phenology Observer, when we offer the volunteers different opportunities for them to interact with our plants, but also helping us with the like in the research department to get the data and we can like analyze it. So to do this, we select and mark the plants that we were interested in. This is the first year, so we like take this as a pilot study or as a pilot like at the beginning point to like kind of to see which like strengths or drawbacks we have from now. And we did a training. We did a training with the volunteers, and then we assigned the plant to each volunteer. So based on the number of plants that we had, we assigned 12 plants to each volunteer, and we gave them the opportunity to visit the plant twice per week and report all of the observation that they have. So we have 23 rhododendron species in total that we are monitoring right now. We have 96 plants in total because we have like 3 replicates per plant. We started this volunteer on April, and they basically do observations around the rhododendron collections, and they record different phenophases, and also we add another part with the pollinator visitors. So they are recording the pollinator visitors that they have or that they can like see in their plants. And all of this is happening at the Rhododendron collections at Holden Arboretum. So I'm going to show like a little bit about the preliminary data. This is basically raw data. I haven't cleaned this because we are waiting for the peak flowering to be done in order to start, looking into like deeply in this data. But a couple of weeks ago, we have 890 reports so far, which is really great. And it's like massive amount of data that we have from the volunteers. They record the phenophases for the plants. We have, in this case, we have like 7 phenophases, and they can report the plants. We already know like the ID from the plant, so we know which plant they are looking at, which frequency do they have looking at the plant, and we also have pictures that we can basically corroborate the information that they are like recording. I also wanted to show a little bit about like the species that we are monitoring and how is the proportion of observations that they have. As I mentioned, this is raw data. We need to like double check and like clean the data, but it's really exciting for us to have like this amount of data and how they, for some of the plants that are already flowering, how they have been reporting like all of the phenophases and how we can in the future, we can interact this with, we have like a monitoring weather station at Holden that we can relate those with the phenophases and the phenology kind of like cycle of the plants that we are monitoring. So this is really valuable for us, but also for the species that we are looking or we are interested in. These are pictures, and as you can see, they are really good taking pictures. So we are also intercepting our work with the education department in order to, so at Holden, they do have, every single plant has a GPS location, and you can basically find all of the information for each specific plant that they do have. And we wanted to include our data in their databases in order to complete the phenology and phenophases data. So we are going to share the data with them, and they basically can use it to use in outreach and like education department, but also for the horticultural department. So we are really excited and happy to have all this data and how we can use this data in any other departments at Holden. As I mentioned, they also record pollinators. So we have a couple pictures here. We haven't, this year is the first year that we do this monitoring. So they are just reporting like the pollinator that they see during their monitoring, but we are trying to establish next year, like kind of like specifically period of time that they are going to sit and watch their plants and basically report who is visiting their plants. So we will have like a better understanding of the pollinator visitors and how this can be related with the other traits that I mentioned before. So to end it up, I just want to mention a little bit about a collaboration that we have with my advisor, Dr. Juliana Medeiros, and other collaborators around. And we are trying to show how these new strategies can reduce the time and enhance or like increase the amount of data that we can collect to make this monitoring and to make like our data or like research more efficient, and how these citizens data can be really useful to, to complete and monitor more number of plants, but also like in more times or more frequently. So as I mentioned, we are using this tool. It's called like Cobo Toolbox. When they can report the data, it's really easy to download. It's really available and free for everyone. And also, I want to highlight the importance of also like iNaturalist and the citizens data that we can or that we have available or already. But also there is a project, and I'm gonna talk a little bit more about this during the roundtable later, and how they are also using camera traps to like kind of monitoring phenology. This picture is from a project in Russia when they were monitoring a tiger, but the tiger was really like closely related with one rhododendron species, and they found that when they were trying to catch the tiger, they had like tons of like recordings of videos, and they saw that they have this kind of like detailed information about the rhododendron phenology, and they extracted around like 20 phenophases from the videos that they are using basically. So I just want to highlight the importance of citizen data and other strategies that can help us as a scientist to reduce and to enhance our data and yeah, the monitoring in the rhododendron species. And I want to thank everyone here, my advisor, Juliana Medeiros, our collaborator, Elliot Gardner from CWRU, and the lab team members, and also the volunteers for all of their work, the hard work that they have been doing. Thank you. I'll take any questions.
Thanks, Catalina. Raise your hand, ask in the Q&A, ask in the chat. We do have more coming up in the roundtable about the pollinator or about the phenology and pollinator monitoring. So stay tuned for that. Catalina, can you just talk a little bit more about the kinds of information you had to give to the volunteers in order for them to be successful in helping with this project?
Yeah, so yeah, I feel like the successful of the program is basically the training that we need to give them. So we provide different materials, so we mark the plants, we gave them like the training was like going into the gardens and provide them with the locations. We created a map that they are using with every single like plant mark or like the GPS coordinates for them. We provide them the form to fill up and how to do it and all of that. And also I created a document that contains all of the phenophases and how to identify them. So basically it's like every single thing that they need to do in detail. But this has been really successful for us, and we think that this can grow up in the next couple of years.
And
In
we're
terms
ready
of
to share it too.
yeah, exactly. Yeah.
Okay, you have two questions in the Q&A. First from Michael Bamford. Does this data collected on site, and what tools are you using?
Thank you. Yeah, this is yeah, in situ or like on site, specifically for the volunteers, right? So they go out in the gardens and they do like walks, and they basically go and check their plants at least twice per week. So they are not collecting any morphometric data. They are just like collecting the phenophases and the phenology data. So they can, with the app, it's really easy to take the pictures, to we create like ID for each plant. So it's really easy to like report the ID. And they do it like, yeah, basically on site.
Michael has a follow-up question on phones.
Yeah, so this app that I mentioned there is like, and I will like mention like any other like apps that they do have in the roundtable, but right now we're using Cover Toolbox, so it's really cool because in real time you can receive the basically the reports that they are doing. You can download the data as CSV, so basically you clean the databases or like the Excel sheet, and then you are be able to use it or analyze it. So it's really good. They do it on like yeah in real time on their phones, or they can basically record the data and go back home and then report their observations. But yeah, in real time they can do it.
And I think Michael's probably thinking about the Rhododendron Society people and whether they might be able to participate. I think the answer is probably going to be yes, but we need a little bit more time to develop it out so that we can manage how we're going to use the data and then make sure our data collection is correct. And I think Catalina Jean has a question about that. How will you consider variation across sites? So things like variation in light and temperature in your approach to analysis.
That's in terms of the VOCs. I mean, okay, so all of the plants that we have are in one location, so the variation that we will have may not be, I mean, that hard to, it won't be enough heterogeneity, maybe. But if we see, we can take like any other approach, like taking temperature inside, or like the plants with thermal cameras, or doing other strategies that we can take, like more like, like more like, I don't know, like small scale data kind of. But the weather station that we have is really in the garden. So I feel like kind of like the scope, or like the data we have is good enough, or we can also use that as a covariate, or something that we can include if there is a high variation in our data among the beds, for example, because the only like distribution is like beds that we can include as a covariate if we saw a lot of variation in when we get all the data.
Awesome. Thank you, Catalina. I think we're going to
Thank
move
you.
on to the